Welcome back, children. Today I'll be taking the different types of cell division. We are going to analyze the differences between the two types of cell divisions, namely mitosis and meiosis. In the same chapter, cell cycle and cell division. The two types of cell divisions are mitosis and meiosis. The first and foremost difference is where it occurs. Which cell does mitosis and meiosis occurs? Mitosis occurs in the somatic cells. What are somatic cells? Body cells. So mitosis occurs in the somatic cells or body cells, whereas meiosis occurs in the sex cells. What are the sex cells? They are the gametes. Second difference, mitosis results in two daughter cells. So as a result of mitosis, two daughter cells are produced, whereas in meiosis, the number of daughter cells that are produced are 4. Third difference, the number of chromosomes in the daughter cells in mitosis is same as that of the parent. If suppose the parent had 10 chromosomes in the cell, I told you the second difference, it produces two daughter cells. Each of the two daughter cells gets 10 chromosomes. So in this schematic representation, I have shown you mitosis, that is from one parent cell we get two daughter cells and the chromosome number is same as that of the parent. In this context, mitosis is also called equational division. So another name for mitosis, equational division. The reason is that the number of chromosomes in the daughter cells are same as that of the parent cell. Now coming to meiosis, if this is the parent cell, the end product, we get 4 daughter cells. That was told in the second point. Now same here, I am going with 10 chromosomes. What will be the number of chromosomes in the daughter cell? Just half. So each will have 5 chromosomes. Okay, so the third point, the number of chromosome is half of that of the parent cell. So that's why meiosis is also known as reductional division. In context to the third point, it is getting another name, reductional division. So third point is the number of chromosomes present in the daughter cells. In mitosis, same, and therefore mitosis is also known as equational division, whereas in meiosis, the number of chromosomes becomes just the half, and therefore it is known as reductional division. What I have represented here is the second and third point in schematic representation. From the schematic representation, you can get 2 points. Your 2 daughter cells and the chromosome number is the same. There you get, from the schematic representation, what you can elicit is 4 daughter cells are produced as a result of meiosis and the chromosome number is just the half of that of the parent cell. Next, fourth point is dealing with the duration, the time span of mitosis. It is short compared to meiosis. So, in mitosis, the duration of the cell division is short, whereas meiosis is lengthy or very long. The reason is that it comprises a number of stages and substages. So let's analyze the fourth point in the next slide. So we are analyzing the different stages of mitosis and meiosis in the following slide. The phases of mitosis are prophase, metaphase, anaphase and telophase. So these are the 4 phases of mitosis. Hence mitosis is very short. The duration is shorter. Clear? Because it just comprises 4 phases: prophase, metaphase, anaphase and telophase. But meiosis, on the other hand, has 2 subphases. So meiosis has 2 subphases, namely meiotic division 1 and meiotic division 2. Okay, meiosis 1 and meiosis 2. And meiosis 1 is further divided into, like we have mitosis, same 4 divisions: prophase, metaphase, anaphase and telophase. But it is indicated with the Roman numeral 1. prophase 1, metaphase 1, anaphase 1 and telophase 1. I'll complete it. And meiotic division 2 also comprises the same stages. They are prophase, metaphase, anaphase and telophase. But here you have to specify with the numerical, Roman numeral 2. So, prophase 2, metaphase 2, anaphase 2 and telophase 2. See, meiosis divided into two subphases, meiotic division 1, meiotic division 2, and each of them are further divided into stages PMAT1, that is prophase 1, metaphase 1, anaphase 1, telophase 1. And meiotic division 2 is again divided the same way with the specification of numerical 2. Prophase 2, metaphase 2, anaphase 2 and telophase 2. Again it is not over. Prophase 1 is further divided into substages. 5 substages. They are leptotene, zygotene, pachytene, diplotene and diakinesis. So once again, prophase 1 is further divided into 5 substages. They are leptotene, zygotene, pachytene, diplotene and diakinesis. So you saw it is very elaborate and hence lengthy in duration. So once again, meiosis have two subphases, meiosis 1, meiosis 2, and meiosis 1 has 4 stages, prophase 1, metaphase 1, anaphase 1, telophase 1, and of these 4 substages, prophase 1 stage has 5 substages, namely leptotene, zygotene, pachytene, diplotene and diakinesis. Now moving on to meiotic division 2, it has 4 substages, 4 stages, prophase 2, metaphase 2, anaphase 2, telophase 2. So this is the complete cycle or the complete phases of meiosis. Getting back to the difference, the previous slide. Now the fourth difference is done. The fifth, the daughter cells in mitosis, the daughter cells is exactly similar to that of the parent cell, very much similar. Whereas in meiosis, the daughter cells are not at all identical to the parent cell. They show variation. How did this variation arise? Variation arises because of a process called crossing over, which occurs, now itself I will tell you which stage, pachytene stage of prophase 1 of meiotic division 1. So that particular phenomenon occurs where this exchange of parts and hence results in variation. Clear? So these are the 5 main differences between mitosis and meiosis. Once again a recap. Mitosis occurs in somatic cells, meiosis occurs in sex cells or gametes. Mitosis results in 2 daughter cells, whereas meiosis occurs in 4 daughter cells. The number of chromosomes are same as that of the parent cell in mitosis and hence the name equational division. Whereas in meiosis the number of chromosomes is just the half of that of the parent cell and hence it is also called reductional division. The duration of mitosis is short. It has only 4 stages, prophase, metaphase, anaphase and telophase, whereas the duration of meiosis is lengthy with an elaborate stages and substages. They are broadly divided into 2 phases, meiotic division 1 and meiotic division 2, and meiotic division 1 is further divided into 4 stages: prophase 1, metaphase 1, anaphase 1 and telophase 1, and prophase 1 is further divided, that stage is divided into substages, 5 substages: leptotene, zygotene, pachytene, diplotene and diakinase. Moving on to meiotic division 2, it's divided into 4 stages: prophase 2, metaphase 2, anaphase 2 and telophase 2. And lastly, the fifth difference is the daughter cells are very much alike in mitosis. They look very much similar to the parent cell, whereas in meiosis, the daughter cells that are formed does not look identical to the parent cell. They show variation, and this variation arises due to the phenomenon called crossing over. Clear? Now we are going to learn the significance of mitosis and meiosis. What is the significance of mitosis? It helps in the repair of tissues. So the first important significance is it helps in the repair of tissues. See, any tissues after operation or bruises or cut or injury, what happened? The repair is taking place because of the cells in the adjacent region start dividing. Then replacement of old or dead cells. We know the RBCs are replaced after 120 days. How it is replaced? By mitosis. Then growth of the body, our bones grow, skin grow, muscles grow. Okay, so as we age from year to year, our body grows. So these tissues also have to grow. They grow by the cell division called mitosis. So once again, the 3 significance of mitosis: repair of tissues, replacement of old or dead cells, and lastly growth of the body. Next, moving on to meiosis. The significance of meiosis: it maintains the chromosome number of a species. See, the chromosome number of human beings are 46. How it is maintained? Because of meiosis. This will be clear when we deal with the chapter reproduction. Anyway, I'll just give a briefing on that. The sperm carries 23 chromosomes. The egg carries 23. So these fuse. When 23 and 23 fuse, when the cytoplasm fuse and the nucleus fuse, what happens? 23 plus 23, we get 46. So the individual form will have 46 chromosomes. So how did we get back that 46 chromosome, or how is the chromosome number maintained in this species, Homo sapiens? The reason is that the gametes that were formed by meiosis, and we know by meiosis what happened to the chromosome number. It gets reduced to half. So the chromosome number of the parent cell was 46. So the chromosome number of the parent cell is 46. What happens? It undergoes meiosis. And we know meiosis, how many daughter cells are produced? 4 daughter cells are produced. 1, 2, 3, 4. And what happens to these daughter cells? So these daughter cells are called gametes here. In this case, it is gamete because it's only applicable for gamete formation. So whether it be the male gamete is sperm and the female gamete is egg. So whatever it be, maybe the male gamete or female gamete, whatever it represents, these are the 4 sperms or 4 eggs. What will be the chromosome number? It will be just 23. Half of 46 is 23. Okay, so like assume this is the sperms. These are the sperms. Similarly, in the female also, gamete formation is by meiosis and the eggs are produced, 4 eggs with 23 chromosomes. Okay, because it is by which division? Meiotic division or meiosis. Now what happens during fertilization? The egg and the sperm fuses. The egg 23 fuses with the sperm 23 and we get back 46. So how was the chromosome number of the species maintained? Because of meiosis. I hope you got the concept clear. And next important significance of meiosis is, we told about variation. Okay, it results in variation. This variation is very important for the process of evolution. This variation you have learnt in 10th standard. Bacteria can survive in any type of environment, right from the hot thermal vents to the cold ice caps. So a wide range of habitat bacteria is found. Why? The reason is because of variation. In any population there are these variants. Okay, and when the condition changes, what happens? The sensitive forms all die, but there are the variants. These variants may be resistant and they will survive. This again you will understand when I deal with the chapter on evolution, where we emphasize on variation enabling evolution. Okay, so remember, variation enables the continuation or survival of the species over time. Even if there is natural or man-made disturbance, because of variation there is maintenance of the particular species. Okay, a very simple example is a man-made. We apply DDT or pesticides to bring down the mosquito population. Initially the population declines, but later you can see the population is re-established. Why? That initial population had two forms, two variants, sensitive and resistant. When DDT was applied, the sensitive all died. There were one or two resistant forms. They mated and multiplied and maintained the population. Okay, when DDT is not applied, what happens? The resistance does not have the survival advantage. They will be less in number, but the sensitive forms will survive. This is the same as the red and green beetle we have studied in 10th standard. Anyway, variation is very necessary in the course of evolution. Clear? So, once again, the last point is dealing with the significance. The significance coming under mitosis is it helps in the repair of tissues, replacement of old and dead cells, and also helps in the growth of the body. This is the significance of mitosis. And meiosis, very two important significance. One is the maintenance of chromosome number of a particular species, and lastly, variation is important in the process of evolution. Clear? I hope you will note down these 6 points and learn it as it is. Thank you.